So today we'll be continuing with the cytogenetic methods. So far we have discussed about karyotyping. We have discussed in the last session on the fish technique, fluorescent in situ hybridization, right? So we have discussed that much so far. This will be the last session for cytogenetic method. We'll be talking about the banding techniques. We'll be talking about other karyotyping, special karyotyping technique, and M-fish technique and CGH method. CGH I think I've introduced in the last session. So yeah, till here we have discussed. So we'll be starting with the special karyotyping and multiple fish method. Now here this technique basically is talking about the ability of isolating one's individual human chromosome using a technique of flow cytometry, wherein we combine the knowledge of human genome sequences, allowing cytogenetics to develop a 24 color probe which sets that this is a whole set of 24 color probe which are used to label the human chromosomes with a very distinct color. Okay, so in this technique we basically isolate the chromosome and develop a whole set of palette of different colors which will be therefore used to label the chromosomes with a distinct color. Similar to the karyotyping and the fish technique, we know that the DNA sample is collected and then it is pooled, and then there is a subsequent hybridization process, right? So here also we'll see that the labeled DNA probe are then pooled and they are used in the hybridization experiments with the metaphase stage of chromosome. Therefore, the labeled DNA probe, it binds to the complementary chromosome, which is to be labeled with a specific fluorescent color along with its entire length. So this is different. Its entire length is where it binds to, and that fluorescence is therefore depicting a certain form of color. It's a very powerful approach understood, which permits the simultaneous tracking of all the human chromosomes, that has been called special karyotyping, or it is also called as multiple fish method, right? So these are the most significant development so far we consider in the field of molecular cytogenetics in past few decades. The special karyotyping method or the multiple fish method, both there's the different names of the same technique. They help us to detect small chromosomal rearrangements in individuals with seemingly normal karyotypes, and therefore determine more precisely the chromosomal aberrations in individuals with complex aberrant karyotypes. So for instance, there's a deletion, but you do not know what exact length of that chromosome, so that deletion is basically it could be difficult to identify if you generally see the karyotype just on the basis of their size and you arrange it on the basis of the size. So identifying that deletion or translocation for that matter, therefore these technical complexities are therefore very efficiently dealt by this technique. Okay, now coming on to the comparative genome hybridization. So I think I've introduced you to this technique. Basically this technique permits the detection of chromosomal copy number changes without the need for cell culturing, because here we are going to talk about it. It gives a global overview of the chromosomal gains or losses throughout the whole genome of a tumor. Okay, so this is the different different feature of this technique, that here we are using the whole genome, and therefore there is no point of doing a culture or pulling out the DNA. So that step is therefore different here. Therefore, CGH is a relatively fast screening technique which can point out at a specific chromosomal regions that might play a role in pathogenesis or say progression of tumors or for instance, you know that a person is having certain gene which could cause tumors. So over the period of time you can conduct a longitudinal study to see and observe whether there's a progression of a tumorous symptoms or tumor in a person, and that diagnosis, therefore that progression over the period of time could be constantly monitored. Okay. Basically this technique aims to quickly and efficiently compare two different genomic DNA samples which are also arising from two different sources, right? So that is also one efficiency of this technique. Originally developed for the purpose of identifying the differences in chromosomal complements of a solid tumor and the normal tissue. So definitely if you have a predisposed gene, so you can identify the differences in the chromosomal structures or its characteristic trait based on whether comparing the two different samples with respect to taking a normal tissue, normal cell culture, and then comparing it with respect to the solid tumor that you are that's your target you're targeting for the purpose of your study. So overall, guided by the CGH results, the more specific molecular biological techniques, for instance, you can go by FISH technique or then heterozygosity analysis and sequencing. All these can be used to identify the oncogenes or the tumor suppressor genes in these regions, right? In this approach, we involve this process involves the two steps. Firstly, the isolation of the genomic DNA, and then fragmenting the genomic DNA from taking the both cases. You take a control subject and then you take a experimental subject. So like the reference we were giving in the previous slide, taking a normal case and taking with respect to a tumorous tissue, right? So this fragmented DNA, therefore, or the fragmented control DNA is labeled with a green fluorescence, and the experimental one is being labeled with a red fluorescence. Then altogether they are pooled and used together as DNA probes in hybridization experiments with the normal chromosomes. So in this case, when there is an unaltered chromosomal regions, both the green and the red probe will equally bind, resulting in some orange or yellow color if they are equally binding throughout the genome. But in case if there's a chromosomal region that was deleted, for example, then it will appear that there are more green under that microscope, and likewise if that region is more amplified, like instead of deletion there is addition, so that region is being amplified, so therefore that region will appear more red under the microscope. So that is how you identify based on this fluorescence technique whether also when altogether they are pooled with respect to the fluorescent dye they are given. Altogether they are pooled, and then based on where they bind to the complementary sequence, if at all they bind or do not bind, so depending on that they exhibit a graphical representation where we'll see what kind of color or what kind of dye is being more represented. So if it's more green color, then you see that there's a process of there's a condition of deletion in the chromosome, and then when you see there's more red color in the graphical representation, you see that there is more amplification of that particular region. So hence researchers can scan along the length of chromosomes to identify these genomic alterations. Okay. The other technical issue here is with respect to this technique that it is very labor intensive method. You have to run it along your human genome to identify throughout your genome like the regions across your human genome, right? So that is very intensive method. Uh, Also, it requires the use of metaphase chromosomes only, and to some extent there's a limited resolution of the regions that we are able to detect at the end, so that are few technical issues associated with this technique. Okay, so far this is about CGH. Now coming on to the banding techniques, which is the most important thing, because ultimately when you're talking about the staining, you're talking about the fluorescence. This technique is basically taking an underlying principle of all these techniques. You have studied in fish also, you have studied in karyotyping also, you have studied about the staining in CGH also, we are studying the staining, fluorescence. So all these things are basically associated with these banding techniques. So it is very important part of cytogenetics, karyotyping and the banding techniques. Okay, so when we know these chromosomes, basically these are appearing to be like thread-like structures, right? They are found in the nucleus of the cell, where you have the DNA, and DNA is basically carrying the genes which are important for your transmission of important hereditary information, right? Or the genetic information, so to say. So when we're talking about these techniques, chromosome bending techniques, these bands are basically, they are identified as parts of these chromosomes which are clearly distinguishable from its adjacent segments by appearing either darker or lighter in shade, depending on what kind of banding technique we are using, right? Then we have chromosomal banding. How do you define chromosomal banding? Basically, these are patterns which are again in lighter or darker stripes. These are appearing all throughout the length of the chromosomes once they are stained with a particular dye. If you see the picture, it more or less appears like a barcode that you see on any product to see that product related information. So it more or less appears like a barcode, and therefore an individual code to one particular genome. So these unique banding patterns are used to identify each chromosome and diagnose the chromosomal aberrations, including whether there's a chromosomal breakage, if there's a loss, duplication, inversion, translocation, whatnot. Every form of chromosomal aberrations could be easily identified once they are stained and they are depicting certain form of pattern. So therefore these patterns, using a specific technique, banding technique, is therefore identifying helpful in identifying the chromosomal aberrations. So that was the whole purpose of doing the studying these techniques, because we are hereby studying the methods to identify the genetic diseases, right? Okay, so this is a very clear and a very easy diagram that I could find on Google. So this here represents, just to show you how that lighter and the darker bands are clearly visible in the as a banding form. So therefore these banding techniques or the staining techniques, I mean, you use that word synonymously, banding or the staining technique for chromosome. Here we will see that both the arms of the chromosomes P and Q, these bandings are clearly visible, right? Also, one more thing I want to add it here, why we are constantly referring to metaphase stage becomes very important. Because here, see, first of all, the chromosomes are very highly stable during stable as in condensed, in a very condensed form they are present at this particular stage. And moreover, when we are doing the staining using particular dye for any technique, that chromosome becomes clearly visible under the microscope because of its more condensed form during that stage. So that is the reason why we are trying to arrest a cell or a chromosome during the during the metaphase stage, so that we can easily identify, easily study the characteristic trait of that particular chromosome, of that particular cell, sorry, right? Also here, when we are saying that there are two different shades, the darker and the lighter shade, so there are very important thing here to be mentioned, that there are two regions of these chromosomes what we are referring to. First is the euchromatins and second is the heterochromatins. So euchromatins basically are the sections of chromosomes which are very much more accessible for the purpose of transcription or for the purpose of translation of proteins or the chromosomes. So therefore these regions becomes the coding regions of the chromosome, the euchromatins. On the other hand, we have heterochromatins. These are generally understood as the known coding regions of the chromosomes. Therefore, they are called as heterochromatins. What we see generally, the AT rich segments, adenosine and thymine rich segments, are the representative of heterochromatins, and therefore they are darker. Like when you see the stripes, the darker stripes are generally the A and T. Okay, for instance, when we'll be talking about G banding, the darker bands will be having more AT rich segments, and therefore they appear to be more darker in shade on the banding form that we see. And vice versa, when we see the lighter bands are euchromatins, they are lighter in shade and they are more G and C rich regions. Here you do not see the here they are more coding, that's considered as a more coding region of the chromosome, right? Also, just to clarify, the darker ones are called as the positive banding, the lighter ones are called the negative banding. So far this was the conclusion, just to clarify you how you study or how you identify what kind of bands are visible, or what is the understanding of these bands, just to clarify these categorization and these identifying traits. Now there has been different banding techniques identified so far, staining or the banding techniques identified so far. This table I'm putting it here because we will not be studying all of these, but it is important to understand which of the technique was identified first, when it started, and who identified it, just for your purpose of reference anywhere else in academics, or for your own understanding. Just go through who all have been the investigators of these techniques, when was it identified, and also see when we're talking about generally what we see, G banding is the most commonly used staining technique in karyotyping, or any other technique for doing the staining. But it was like lately identified after the Q banding, after the Q necrine dye was used. So just for your understanding why it started, you can go into the history of these techniques if at all you're interested. R banding, which is generally considered as a reverse form of banding of G banding, was very identified very after the formation of G banding. So also, apart from these mainly used Q, C, R and G bandings, there are other forms of bandings like NOR banding is there. Then there is T banding, which are very commonly also used for research purposes and things like that. So just putting it here just for your further reference. Now, what is exactly the pathway of how do you do the banding procedure is here what we are going to talk about. Firstly, here the cells are grown in the culture, right? Unlike any other method, then we'll be trying to arrest the cells when then the cells will be entering into the metaphase, and here is what we are trying to arrest the cells at this particular stage. So to arrest the cells are treated with the mitotic inhibitor like a colchicine. That is a very commonly used inhibitor, and that is how we are arresting the cells at this particular stage. After being arrested, taking the cells at storing the cells at metaphase stage, then those cells are stained with particular dye, depending on which technique we are using. We'll be talking about it. So that chromosome is stained, and after being stained, they'll be observed under the microscope for identifying what kind of banding has been visibly there. Okay. Now, what are the types of banding techniques we'll be covering? Firstly, the G banding, then Q banding, C banding, and R banding. These are most commonly used or referred to or studied forms of banding techniques. So therefore, I'll be sticking to these 4 for the purpose of your academics. Firstly, starting with G banding. Now, G banding is named after, G refers to the GMSA dye that is used for this technique. Firstly, here we are resting the cells at a metaphase stage. Then we are treating the cells with trypsin or urea or protease. Here we are using these enzymes for the digestion of proteins or the chromosomes, right? So that's very important step for this banding technique. Then what we are doing is we are staining the chromosomes with the GMSA dye. The banding obtained are both light and dark, but here the dark bands are A and T rich bases, so therefore there will be darker regions, right? Or you will say them heterochromatins. On the other hand, you will have the lighter bands, which are G and C rich bases, and therefore they will be more euchromatin side, right? So therefore this is how you understand it. Coming on to the next form is the R banding, which is called the reverse banding, reverse of the GMSA banding technique or the G banding technique. Here also we'll first arrest the cells at the metaphase stage, but they are firstly pre-treated with a very hot salt solution, and then the DNA is made to undergo denaturation to get its single strands. Then after that process, after the denaturation happens, the chromosomes will be stained with the GMSA dye. Also here we are using the same GMSA dye, but the darker band in GMSA banding are the lighter bands of R banding. So therefore that is why it is called as vice versa, because here we did the denaturation, so that has impacted on the studying of the bands that we are getting in the R banding technique. So here the darker ones becomes the G and C, which are the G and C rich bases, and the lighter ones become the A and T rich bases. The analysis of the structure of chromosomal ends of these areas are lightly stained than the G banding techniques. Coming on to the third method, which is Q banding. Here the chromosomes are stained with the quinacrine mustard dye, and after being exposed to that particular dye, they are kept under the UV radiations, UV light directly. Then after being stained, after being kept under observations, then we'll be seeing the banding patterns. Here we'll see that dark bands are A and T rich, and the lighter ones are G and C rich. But there's disadvantage associated with this banding. What you see basically, because firstly that being exposed to UV radiations, there are chances of chemical bonds being broken due to that constant radiations. So that could hamper the observations, that could hamper the banding formations, banding patterns. Secondly, over the period of time, this particular dye has a tendency to fade out. So instead of showing the darker bands, it will exhibit lighter bands. So that tendency becomes it has tendency to fade out. That mustard dye has a tendency to fade out. So that could also hamper the observations. On the third disadvantage, we'll see that this dye also is having the capacity or the tendency to absorb these UV radiations, UV light, due to the chemical bond which is being formed between the dye and the light. So that could also again reflect on your banding pattern. So these 3 are very good disadvantages that you can cite. Coming on to the last banding technique, the C banding. The C banding basically means constitutive heterochromatin regions of your chromosomes. Here the stain areas of heterochromatins are very tightly packed, and they have very constantly repetitive DNA. Here also chromosomes are firstly denatured using the enzyme, using the chemical called barium hydroxide. Then they are stained with GMSA dye. Only the dark band is visible in this case, which depicts the constitutive heterochromatins. The lighter bands here are the non-coding regions of the chromosome, and it constitutes of the rest of the region of the chromosomes, right? The main important application that you find of C banding is also in the purpose of chromosomal aberrations identification, but also for the purpose of paternity testing. So these two are very good advantages of it. Now there are other banding techniques. We generally refer to other T banding and the NOR banding, but for the purpose of your academics, we will be sticking to these 4 categorizations only. Okay, but anyhow, this list of techniques is already being cited here, so you can go and look for, if at all you are interested in reading more about the chromosomal banding techniques, right? So this was it. We are going to conclude the cytogenetic method. Next we'll be starting with molecular and the biochemical methods. So if you have any queries, just let me know. Okay, thank you.